For this next chapter, we're going to talk about genetics, evolution, development, and plasticity. So when we speak of your genetics, we are talking about your nature, your genes, and how your genes interact with your environment to shape who you become. One of the biggest issues that exist in human life is how much of a role genetic or environmental factors play in human behavior. When we speak of your genetics, we're talking about anything related to your genes, facial expressions, psychological disorders, weight gain, personality, sexual orientation, etc. In the 19th century, the monk Gregor Mendel said that people inherit genes through their families, and it happens in discrete units. Before Mendel, people believed that inheritance had to do with blending of the egg and the sperm, like paint. But he said, no, it happens through units of heredity. The genes that he said come in pairs are called alleles, and they're aligned along the chromosomes. So a gene is defined as the portion of your chromosome and is composed of DNA. DNA is what serves as the model for synthesis of RNA, which is a chemical that serves as the template for protein production. All of these things you need in your body to help you function regularly on a daily basis. Proteins determine the development of the body by forming the structure and serving as enzymes that help regulate the chemicals throughout your body. Homozygous genes are the genes that are identical pairs of genes on the two chromosomes. So homozygous are going to be the same, identical. Heterozygous genes are the genes that are unmatched pairs on the two chromosomes. So they're different. And a lot of this should seem like basic biology. When it comes to your genes can be either dominant, recessive, or intermediate. A dominant gene is going to be a gene that shows a strong effect. It's dominant. It's clear. It can be either homozygous or heterozygous in condition. A recessive gene is a gene that's going to show its effect only in a homozygous condition. An intermediate gene is going to be a gene that occurs in a phenotype where there is independent dominance in the heterozygous condition. So if it's dominant, it can be homo or heterozygous. If it's recessive, it's going to be homozygous. If it's intermediate, it's going to be heterozygous. And this is all according to Gregor Mendel. There are certain conditions that can occur, such as PTC or hair color, and PTC being a taste sensation, and hair color, which can be very misleading because they imply that a single gene combination controls the characteristic, but it could be something else. Some genes are only partially expressed in some cells and not in others. So lots of talk about genes. An autosomal gene are all the genes except for sex-linked genes. And the sex-linked genes are different. They are related to only the sex chromosomes. Females have 2 X chromosomes. Males have an X and a Y chromosome. During reproduction, females contribute an X chromosome all the time because that's all they have to give. And males will either contribute an X or a Y chromosome, and that is going to determine the sex of a child. So if a male contributes an X, then it will be a female child. If a male contributes a Y, then it will be a male child. The Y chromosome has genes that have far fewer proteins than the X chromosome. Therefore, sex-linked genes usually refer to X-linked genes, like red-green color deficiency. So the Y gene just doesn't have as many proteins for that. Sex-linked genes are also genes that are present in both sexes but have an effect on one sex, like chest color, chest hair, breast size, etc. Genes are able to mutate, which is a heritable condition in a DNA molecule, and that means it can spread. If you have microduplication or microdeletion, that means that part of a chromosome that might appear once might appear twice or not at all. If it's microdepletion, it might appear twice. If it's microdeletion, it might not appear at all. Many researchers, when it comes to the field of psychology, believe that schizophrenia might be the result of microduplications and microdeletions of brain-relevant genes. There's lots of research on the cause, but we know it is inheritable. The field of epigenetics is a field that's concerned with changes in how your genes are expressed without the modification of your DNA sequence. So some genes can be active only at a certain point in a person's life or even at a certain point in a person's day. And then changes in those gene expressions are central to learning and memory. Because of epigenetic differences, people are likely to explain the differences between identical twins and fraternal twins. We know that identical twins are genetically the same in terms of DNA, whereas fraternal twins have the same DNA as any other brother and sister, even though they're born on the same day. Anything you do at any moment affects your genes. It can impact your epigenetics. When it comes to heritability, this refers to how much of the characteristics a person has depends on their genetic differences. Researchers have found that there's evidence that heritability in almost every behavior has been tested, and that different behaviors can be inherited from families. Heritability of a certain trait is specific to a given population, and we know that there are strong environmental, so nurture, influences that may cause genetic influences as well. So if a person is raised in a different environment, that can also impact how their genes are expressed in the future. Again, when it comes to nature and nurture, the nature and nurture debate in psychology is a big one. It's always existed. It probably always will. We do know that most behaviors have a genetic and environmental component. And the way we know that is because there's been many studies of twins that tell us that. Because all you have to do is take a set of identical twins, raise them in different environments, and see what differences exist. Many adopted children resemble their biological parents in many ways, and we know that they can have some of the same behaviors of those parents as well. The environment can also impact how things are metabolized, such as the condition PKU, which is a genetic condition that can exist in children, which has to do with amino acid expression. Genes themselves do not directly produce behaviors, but the proteins produced by the genes increase the probability that a behavior will occur. So it's not the gene that causes the behavior, but it's an interaction with the proteins and the genes that can have an effect. The genes can alter your environment by producing behaviors or traits that alter how people in your environment react to you. So over time, everybody's behavior has evolved. Evolution refers to the change in the frequency of genes in a population over time. It answers two questions. How did some species evolve, and how do species evolve? When we think about evolutionary theory, the evolution of species is based on offspring resembling their parents, gene mutations, and certain people reproducing more than others. Artificial selection occurs when people choose individuals with desired traits and make them the parents of the next generation. So it's like the parent who decides, okay, I'm going to have artificial insemination, but I want to have a child who has this color eyes and this color hair and things of that nature. That will impact future generations. According to Charles Darwin, who we know is the guru of evolution, nature, a person's nature, their genes, can select how successful individual genes will be prevalent in future generations. Lamarckian evolution refers to the use or disuse of some structure or behavior which causes an increase or decrease in that behavior. In some ways, humans have stopped evolving and improving. When we think about your brain and the evolution of your brain, we know that our ancestors were able to get enough nutrition to provide them with a brain that helped them function. They were able to cook food that made it easier to digest. They were able to hunt, which was efficient. And humans now have a better capacity for glucose transport because of all those things that happened long ago. The field of evolutionary psychology focuses on how human behavior has evolved over time. It assumes that human behavioral characteristics have evolved through natural selection and survival of the fittest. Things that helped a species survive over time have continued, whereas things that did not help a species evolve have been discontinued. Lifespan length, gender differences in sexual promiscuity, helping behaviors or altruism are some of the behaviors that have been more debatable in regards to natural selection. And if you think about it, it's probably because some of this stuff may have an environmental component more so than a genetic one. One gene spreads if individuals with it reproduce more than individuals without it. That can benefit the species, but not the individual who dies out with that individual. That individual trait is going to die out with that person. Kin selection has to do with the selection of a gene which benefits an individual's relatives, and group selection can be controversial because it is the idea that altruistic groups or helping groups survive better than less cooperative ones. And in American society, that certainly is controversial because this is the difference between living in an individualistic society and a collective society. And America is a very collective society where it is all about competition. I'm going to take care of me and mine versus necessarily let me help all of mankind. Reciprocal altruism is the idea that individuals help those that will return the favor. It's like tit for tat. You do for me and I will do for you. When people build a reputation of helpfulness, it's only going to work if others are willing to cooperate with them. And we know that there are many people in our society who are very helpful and like to help and do for other people. As your brain develops, maturation and learning are two things that helps your brain receive the power it needs. Learning can impact how neurons develop, how axons connect, and it can modify your genetic development. When it comes to learning, we learn what behaviors we need to do and continue in the future and what behaviors are not beneficial to us. In the human nervous system, it begins to form whenever an embryo is created at about 2 weeks of gestation. If you take a look at the picture here, it can kind of show you when a baby is an embryo at about 3 weeks. Notice their central nervous system is already starting to form. And then by 7 weeks, it's forming more. By 11 weeks, it's a lot more formed. And then at birth, it looks a lot like it will look in adult life. Cerebrospinal fluid is a fluid that is central in your spinal cord and the ventricles around it. When a baby is born, their birth weight is approximately 350 grams. By the time they are in their first year, their brain is approximately 1,000 grams, and an adult brain weighs 1,200 to 1,400 grams. Neurons in your brain developed through these 5 processes, and we're going to talk about each of them in detail, starting with proliferation, which is the production of new cells and neurons in the brain early in life. These cells line the ventricle divide. Some become stem cells that continue to divide, and others remain where they are and become neurons or glia that go to other locations. So proliferation has to do with producing new cells and neurons in the brain when you are in early life. Migration is just like what migration means everywhere else. The movement of newly formed neurons and glia to their eventual locations. Some reach their locations and stay until adulthood, some do not. Differentiation has to do with the forming of the axon and dendrites that give the neuron its definitive shape. So it's differentiated, okay, it's now becoming different. It's starting to look like something. And that axon will grow first, either during migration or once it's reached its target, and then the dendrites develop later. Myelination occurs when glia produce that fatty myelin sheath that covers the axon after it's been developed. It's very important for that myelination to occur because that is what's going to help speed up the transmission of a neural impulse. This, of course, is going to first occur in the spinal cord, and then it goes to the other areas of the brain. Synaptogenesis is the final stage of neural development. It is when the synapses are formed between the neurons, and we need those synapses to be there so that neurons can communicate with each other. This also can occur throughout the lifespan, and it tends to slow down as you get older. So most people don't think that as you age, you can develop new neurons. However, later research has shown us through stem cell research that there are undifferentiated cells that are oftentimes found in interior parts of the brain that can generate what are called daughter cells. And those daughter cells can transform into glia and neurons. Also, olfactory receptors can be regenerated to replace the ones that have died over time. Development of new neurons can happen in other parts of the body as well. Stem cell research has told us that new neurons in the hippocampus of mammals can help facilitate learning. In your life, some neurons are able to survive more than others. Most neurons have an average lifespan, and some of them are under a year old, like your skin cells, which are the newest because your skin is ever evolving. It's probably the most exposed part of yourself. Your heart cells are probably as old as the person because you always need your heart to survive, and you want those neurons to continue to develop the way they're supposed to. Axons are able to travel great different distances across the brain. In 1954, the researcher Sperry said that axons follow a chemical trail to reach their target. These growing axons are able to reach their target, and because they follow chemicals that are attracted to other chemicals and not some. So when it comes to your axons regenerating or developing, there's a general principle that we have to follow. Axons are going to reach their targets and the target they form synapses with. Postsynaptic cells can strengthen the connection of cells and eliminate others. And the formation or elimination of these connections depends on the pattern of incoming information. Neural Darwinism is the belief by some theorists that the selection process of neural connections exists. That differs from the idea of synaptic connections. More successful axon connections and combinations survive, while others fail to sustain active synapses. So researchers have been going over the idea of how things work in your body for many years. And to be quite honest, they don't really know which has more of an impact. Levi Montalcini discovered that many muscles do not determine how many axons form. They determine actually how they survive. So the term nerve growth factor has to do with a protein that's released by your muscles that promotes the survival and growth of axons. Neurotropins are chemicals that promote the survival and activity of neurons so that those neurons will continue to exist. Axons that are not exposed to neurotropins after making a connection go through apoptosis, which is cell death. So our healthy adult nervous system contains no neurons that fail to make the appropriate connections. We want to keep healthy neurons so that our body continues to function appropriately. Neuronal death has to do with the elimination of neurons that are unhealthy for us. After maturity, apoptosis occurs and becomes more dominant because it needs to get rid of the things that aren't being beneficial to us. The visual cortex is very thick, and in blind people, because there's no visual stimulus that they have since they cannot see. Your brain is the most important organ in your body, and it can also be very vulnerable. When a child is first born, in the early stages of brain development, that's a critical period. If there's a mutation of a gene, that could lead to birth defects, and chemical distortions in the brain can also lead to developmental problems. That's why we try to reduce teratogens or chemicals or virus or bacteria or drugs and alcohol from a pregnant woman's experiences so that the brain is able to develop appropriately. Fetal alcohol syndrome is a condition where children are born after their mother has had heavy drinking during pregnancy. Children who are born with fetal alcohol syndrome could be hyperactive, they could have trouble maintaining their attention, they could have facial abnormalities and motor problems and heart defects. Their dendrites are short and have very few branches. And having exposure to alcohol as a fetus can suppress glutamate and enhance the release of GABA. And that means that many of their neurons will be less excited and exposed to neurotropins that undergo normal apoptosis. So some of them that need to die won't be able to die. Neurons in different parts of the brain differ from one another in shape and chemical components. Immature neurons develop part of the cortex and the properties in new locations, and neurons transplanted at later stages of development can have some new properties and also retain old properties. The brain has the ability to reorganize itself in response to its environment. Dendrites can grow new spines, axons, and dendrites can continue to modify their structure. We know this from research on animals such as rats. Rats who are raised in an enriched environment can develop a thicker cortex, and they can have a measurable expansion of neurons, and some of their old neurons can die and new ones can form to take its place, which causes their learning and memory to increase. It was once believed that teaching a child a concept like math and Greek or various languages could enhance their intellect. That is known as VAR transfer. But evidence has shown us that the skills associated with practice task transfer, that some skills are associated with that, but others are not. You can't exercise your brain like a muscle. So yes, you can read, and reading makes you smarter, and exposing yourself to new information makes you smarter, but there's no way to exercise your brain the way you would get on a treadmill. Blind people can improve their attention to touch and sound based on practice. We know that when one area of the brain is not working properly, other areas will overcompensate for that. The occipital lobe is dedicated to processing visual information, but can adapt to processing tactile and verbal information. And that's why those senses become stronger. And this is the same thing. People who are blind from birth are better at discriminating objects and touch because they work on that, because they practice that skill, and those senses are able to take over those functioning. A lot of research has shown us that magnetic stimulation in activated areas of the brain, such as your occipital cortex if you're blind, can help people discriminate between tactile stimuli like Braille symbols. And even though people who have sight won't be able to read Braille. MRI studies have also told us that when it comes to learning music and becoming a musician, the right hemisphere is 30% larger in people who are musicians than people who aren't. There's a thick gray matter in part of the brain that's responsible for hand control and vision and being able to use a keyboard. That is because of practice and how your brain is able to reorganize itself to perform that skill. Adults can also modify their brain anatomy just by practicing different things they're similar to. There is the idea, though, that your brain can reorganize itself too much. Focal hand dystonia, or musician's cramp, is a condition where the reorganization of the brain has gone too far. When a musician has practiced so much that they become clumsy, fatigued, and they make mistakes. This can also be the result of extensive reorganization of the thalamus and the cortex so that touch responses to one finger overlap to another. And that just means that your brain can be overwhelmed through too much practice and too much of reorganization of those brain cells. When it comes to adolescents tend to be more impulsive than adults. And that impulsivity, of course, can lead to problems and risky behaviors and discounting of the future. Well, when we think about it, the adolescent brain is not fully developed. They are not equally impulsive in all situations. Some peers make good decisions and some don't. The prefrontal cortex at the front of the brain, which is a part of the frontal lobe, is inactive in certain areas, and that can be a cause of their impulsivity. It may not be. As the brain gets older, neurons lose their synapses and the person tends to slow down a bit, and some of the brain structures lose volume. People who are in older age vary in respect to intellectual. They can be different in respect to intellectual decline, and although they have a better base of knowledge and experiences, those things can compensate for any memory losses or cell death that might occur. People who have gone through brain damage can show remarkable recovery. That is because the brain is plastic and can grow those new neurons and form those new axons and dendrites. Some of the mechanisms that are responsible for that brain recovery include the same mechanisms that are involved in development, such as those axons and dendrites. Here are some possible causes of brain damage: tumors, infections, closed head injuries, various diseases, or exposure to substances. If you take a look at this picture here, this is the brain of a person who died after a stroke. And of course, we can't look at any of these brain structures until death. The second one is the brain of someone who survived for a long time after a stroke. Look at how different it is. And the third one is the brain of someone who suffered a gunshot wound and died immediately. So just look at how those three brains look different. So when a person has had a closed head injury, that can be a sharp blow to the head that does not puncture the brain. This is one of the many causes of brain injury in young adults. Boxing, sports, things like that can cause closed head injuries. After that injury, recovery can be slow and incomplete. A stroke is a temporary loss of blood flow to the brain, and it's a common cause of brain damage in the elderly. There are two types of strokes, ischemia and hemorrhage. Ischemia is the most common type of stroke, which has to do with a blood clot in an artery where your brain loses oxygen and glucose supply. And we know that strokes can also be deadly. And the hemorrhage is a less frequent type, resulting from a ruptured artery. And this is when your neurons are flooded with blood, calcium, oxygen, and other chemicals. So after a stroke, a person could have edema, which is fluid that results in the brain and causes increased pressure, which will cause other strokes to happen in the future. And there could also be the disruption of the sodium potassium pump, which could cause issues with accumulation of potassium in neurons. Both of those things could trigger the release of glutamate and overstimulation of neurons and cause other things to happen that could kill those neural cells. So there are some treatments for people who've suffered from a stroke, such as drug treatment, which will break up those blood clots and reduce the effects of the ischemic strokes. And there's also been research that tried to stop those neurons from dying by using drugs. One of the most effective methods is to cool the brain, and that is reducing the amount of overstimulation, cell death, and inflammation. Marijuana has also been shown to help potentially minimize cell loss after a brain stroke, and it has an anti-inflammatory effect. It's most effective before a stroke, but can be effective after a stroke. So if a person has suffered from brain damage, those surviving brain areas increase or reorganize their activity. Drugs can stimulate the activity of the healthy brain. Destroyed cell bodies cannot be replaced, but damaged axons can grow back under certain conditions. For example, if the axon in the peripheral nervous system is crushed, it follows its myelin sheath back to the target and grows again. So when an axon is able to regrow, it does not readily regenerate. It takes some time. It has to have building protein bridges that can help. Axon sprouting has to do with the sprouting of new branches formed by other non-damaged axons that attack to the vacant receptors. Those cells lose their structure but are able to release neurotropins that induce axons to form. The nervative supersensitivity has to do with those postsynaptic cells which are deprived of synaptic inputs. This is the heightened sensitivity to a neurotransmitter after the destruction of an incoming axon, which can cause things such as chronic pain or fibromyalgia. When sensory receptors are reorganized, it can cause the symptom of phantom limb, which is where a person continues to feel sensation even though a part of their body has been amputated. That's because those axons that are degenerated leave those vacant synapses and other axons sprout, and the person thinks their body part is there even though it isn't. A touch on the face can bring the experience of phantom arm pain. The use of an artificial limb can reduce the likelihood that a person will experience phantom limb pain. But many people who have lost a limb, such as a leg or an arm, don't want to have an artificial limb in the place of it. Some people function without that. So anytime limbs have been lost and that impacts their sensory input, these things can still be used, but are often not used because other mechanisms in the body can carry out those behaviors a lot easier. This has led to the development of therapies that improve functioning of brain damaged people. And I know that was a long chapter, but that is everything for genetics.